We are welcoming Robert Bertin tonight as the distinguished speaker for 2021 to the New England Botanical Society. A little bit of background to Dr. Bertin. Many NEBC members know Robert very well, but I'll just give a brief bio here. Robert Bertin obtained his Bachelor of Science degree in biology from Hobart College, and then a Master's of Science at the University of Connecticut, and then went on to do his PhD in biology at the University of Illinois in Champaign-Urbana, finishing in 1980, after which he taught for 2 years each at Bucknell University and Miami University in Ohio. Then Dr. Bertin moved to New England and joined the faculty at the College of the Holy Cross, or Holy Cross in Worcester, Massachusetts in 1984, where he's taught a range of courses from ecology and field botany to environmental biology and biological statistics. Early in his career, Dr. Bertin's research focused primarily on the ecology and evolution of flowering and sex expression in various plant species, and had a number of publications in journals like the American Naturalist, the American Journal of Botany, and Botanical Review. More recently, Dr. Bertin has shifted his focus to think about the effects of recent climate change on flowering phenology and changes in the floristic composition of New England, with work focused on documenting and understanding patterns of plant distribution and how they've changed over time. His research, this recent work, is really exemplifying this powerful intersection of the use of biological collections like herbaria in the region, pairing that with contemporary field work to quantify and understand shifts in the flora of our region. These floristic and phenological investigations have led to a number of notable publications in journals like the Torrey Botanical Society, American Midland Naturalist, and NEBC's own Rhodora, as well as important benchmark publications co-authored with other botanists in the region, documenting the floras of Worcester and Franklin counties in Massachusetts, which he'll be speaking about tonight. Dr. Bertin retired from full-time teaching in the summer of 2020 and is currently Professor Emeritus of Biology at Holy Cross, but plans to continue teaching on an occasional basis, including offering his popular field botany course in the fall. We are really delighted tonight to welcome Dr. Bertin as New England Botanical Society's distinguished speaker for 2021, in part in recognition of his major contributions to documenting and understanding patterns of regional floristic change in our region. So without further ado, I'll hand over the Zoom to Dr. Bertin for his presentation on regional floras and the assessment of floristic change.
Thank you very much, Jesse, and I would like to thank you and the other officers of the club for the invitation to give this lecture. I feel very honored to do so. And I would also like to welcome everyone out there to the talk. I can't see you from here, but I know there are some people out there, and thank you for joining me and the other members of the club. Hopefully, this is one of the very last times we will be forced to do this by Zoom, and it'll be very nice to get back and see you all in person again, hopefully in the fall. Well, floristic botanists by and large inventory floras, and they are particularly interested in spatial patterns of species and also temporal changes in those patterns, and in understanding the factors that are causing both the patterns and the changes in those patterns. And there are a number of sources of information that can be used for these floristic evaluations. And I want to just mention 3 of them here. First of all, you have the published flora. And it turns out that Southern New England and Massachusetts in particular have a very rich history legacy of these sorts of floras, and a few of them are illustrated here. There are also several others. And of course, these provide, in many cases, excellent documentation of the flora of a particular area at a particular time. Some of them also attempt to include a temporal component to them. And of course, this is most practical when somebody else has worked on a flora at some of the same area at a previous time. There are some potential challenges in interpreting changes when you're comparing two floras, because in many cases you don't, particularly the historical floras, did not leave a detailed record of exactly how they sampled. That is, how many hours went into it, how they distributed their time in different areas and so forth. But in fact, the comparative examination, a number of places have had such an examination: Middlesex Fells, the town of Concord, Worcester County, and there have been others. And so this is one very valuable source of information. A second one, obviously, is our herbarium collections. And I would like to just take a moment here to acknowledge the 125th anniversary of this organization, club, now society. And in particular, and thanks to Nancy Eyster-Smith for these images, some of the founding members of the club contributed very importantly to the things that I'm going to be talking about today. And in fact, I think the members of the club in general can give themselves a bit of a pat on the back here, because the herbarium of the New England Botanical Society has the most extensive collection of New England specimens anywhere. And it is through the efforts of numerous individuals over a period of more than a century that have accumulated this record. This is J.F. Collins here with some of his specimens. This happens to be a label of an herbarium specimen of his. This happens to be a collection from Miller's Falls, Massachusetts, which is in Franklin County. Emil Williams, E.F. Williams, also collected in Franklin County. This was a specimen of Pyrrhilla americana, misidentified actually. It turns out is Pyrrhilla chlorantha, but the important thing is you got the specimen, you got the location information, and so forth. Now, today we are very fortunate in that the large majority of New England specimens, the data on them have been placed in digital format and assembled by this group, the Consortium of Northeastern Herbaria. And in fact, this database, which is available online, has over 1.6 million specimens in it, most of them with images, and is a fantastic source of information that did not exist 10 years ago. I should point out that there are errors in it. When you are logging in that many specimens, things are going to get sometimes entered incorrectly. But gradually those errors will become fewer as they get weeded out. So herbaria are obviously a very useful source of information, but they are subject to some very, very pronounced biases, as has been summarized in a number of publications, and I'll actually show you a few slides to illustrate this a little bit later in the talk. New England herbarium specimens go back about 200 years. A third source of information is available for a fraction of our species, and these are data that come from the Natural Heritage and Endangered Species programs in the different states. Each state has a number of rare species, which are the so-called state-listed species. And this is just a screen capture from the website for the Massachusetts Natural Heritage Program. And they have a viewer there, which this is a screenshot of, and you can enter in any species that is listed, and you can pop up a map that shows the towns in which that species has been recorded. And there's also a little table that indicates the dates of the last records for a particular town. So that's a very useful information. It is collected from various sources, including both published floras and herbarium specimens. But there are also additional records that are put in there by the state botanist, Bob Wernerall, and other individuals in his office, and also contributions from other botanists. So those are three sources of information, and I'm going to be dealing with all of those today. There is a fourth source, which I Some people have used, I have not found it that useful, but there are the so-called citizen science platforms like iNaturalist. And you could certainly go on iNaturalist, and if you want, take purple milkweed and look that species up and see what people have posted. I find for the rarer species, sometimes the location information appropriately is not given. And in general, there aren't that many records, but that is a fourth source of information that may be of use for some projects. So that's a quick summary of the sources of information. What I'm going to do in the remainder of the talk is give you three fairly brief sketches of three different projects. Each one of these projects, I might add, is a collaborative project of which I'm just one member. And second of all of these projects are currently ongoing and unfinished. And so what you're seeing here is a kind of a mostly late stage progress report, but things are subject to change by the time, hopefully, they reach publication. So first of all, I want to talk about the, well, let me just tell you what the three are. The first is going to be an analysis of changes in the flora of Franklin County, Massachusetts. The second part is going to be an analysis of changes in rare species in Massachusetts that have northern affinities. The third sketch is going to be specifically about orchids and some of the changes in abundance that have occurred in that family. So first off then, changes in Franklin County. Franklin County is nestled up against Vermont and New Hampshire. It straddles the Connecticut River. This is a project that was worked on principally by five of us, these four intrepid individuals here and myself, with additional assistance from a number of others. And I think if you look carefully at these images, you'll realize that Peter Grimme is the only person taking this project seriously. Okay, just to give you a very quick summary of what we did and what we found. Over 3,000 hours of field work, logging over 46,000 field records. We collected over 4,300 specimens and also examined over 16,000 herbarium specimens and several thousand literature records. And of course, it was the herbarium specimens and the literature records that were used to establish the historical flora. Findings, very briefly, this is just a map of the county, so you can see the towns outlined. There are 26 towns, and this happens to be a plot of species richness in the different towns. And you can see that the towns straddling the Connecticut River, this is the Connecticut River coming down through here, the towns straddling the Connecticut River tend to have the highest diversity, with the two winners being Greenfield up here and Montague down here. In total, over 1,200 native species and over 600 non-native species were observed either in the recent period or historically. If we look among the native species, fairly modest changes. There were 67 species recorded historically that we did not relocate. We recorded 50 native species that had not been recorded historically. And most comparative floras show higher rates of species lost than this, but most comparative floras are analyzing smaller areas, and you'd expect greater species losses in smaller areas. And the other thing is that Franklin County, much of it is quite rural and is less impacted perhaps by changes in land use than would certain other areas of the state be. So let's look at, since we're talking about change, we need to have a numerical measure that captures change that we can calculate for each species. And I don't want to get bogged down here, but in case you're interested, this is how we calculated it. We based it on the town level because the herbarium specimens usually only recorded town. They didn't record more detailed locality information. We started the project in 2010. So any town in which a species had been recorded since 2010 or after was considered a recent town. Anything recorded prior to 2010 was a historical town. And we calculated the change index as the number of recent towns minus the number of historical over the sum of these two. And this denominator, in part, is to make sure we didn't have zeros down there. For example, if we just did it over the number of historical towns and we're dealing with a species that wasn't recorded historically, then we can't calculate the index. The result is an index that will be 1 if a species is recorded only in recent towns, -1 if it was recorded only in historical towns, and 0 if it was recorded in the same number of towns in the recent period and the historical period. So numerically, it's kind of a tractable number. And these are 3 examples of the change index. The symbols you see on the map are keyed here. Elaeagnus umbulatus, the autumn olive, has a change index that is close to 1, actually, no surprise. Lots of pluses here, meaning these were towns where the species was recorded only in the recent period. So we have a species that is showing an apparent increase. Orthilia secunda is showing an apparent decrease. And there are lots of open circles here, reflecting towns in which only historical specimens are known, and the change index is negative. And then this would be an example of a species with fairly little change. Lots of solid dots, meaning the species has been recorded both in the historical and recent period, or you could have simply a similar number of pluses and open circles. Okay, so hopefully that gives you a little bit of a feel for the change index. Now, first of all, let's just look at native versus non-native species. If we look at native species overall, the median change index was 0.18. Now, you might say, well, does that mean native species are spreading to more and more towns? No, probably not. That's likely to result from the fact that we were pretty systematic and thorough in our sampling of the different towns, whereas historically, the sampling was less systematic. And so, in fact, probably that is a species that is not actually changing in its town level abundance would be expected to show a change index that is modestly positive. Well, how do non-native species compare with these? Okay, on the left, we have the same curve that I just presented, or the same bar graph. On the right, we have the plot for non-native species. And you can see that they have a very different sort of change index, much higher median. One possible explanation is that those collecting in the past didn't pay much attention to the non-native species, but there's a pretty good chance that this is, I think, very likely, this is due to actually increase in the prevalence of non-native species. And I'm putting up in the next slide the Franklin County data side by side with Worcester County data collected in a fairly similar way. And you can see a very similar type of trend, okay? Non-native species are increasing relative to native species. This is not surprising. Lots of other studies have demonstrated this. We actually had the society had a speaker a few months ago who argued that botanists pay too much attention to the distinction between native and non-native species, but I would actually have to disagree with this, and I think it's very important that we do pay attention to that, because I think there are good ecological reasons to do so. I'll get to those in the next slide. This is simply a map of 3 species that you probably are, all of which are familiar to you, showing how they have increased in Franklin County. But as far as what the ecological implications of this are, there are now a number of studies that show data of this type. This is a comparison of the abundance and the diversity of insect herbivores on native and non-native shrubs. These are paired by genus. And you can see that the green bar is higher in every case than the blue bar, and in many cases it's significantly higher. So this means that native species are better food plants for herbivores than are non-native food plants, non-native food plants. And of course, insect herbivores give rise to things like this, to tiger swallowtails and luna moths. And so to maintain insect diversity, maintaining native plants is important. And then of course, we have to consider the trophic levels above that, because if you have a low diversity and density of insect herbivores on your plants, that means that there may be less diversity of birds or other organisms that feed on those. So I would argue that attention to non-native species is very important, and we should continue to try to minimize the effects of the spread of non-native species in the landscape. Okay, well, that's not, as I say, lots of other studies have shown similar kinds of things. Let's move on and look at some other questions. A very common question that is looked at in comparative floras is whether there are any habitat-specific patterns. And in this analysis, we recognized 9 different habitat types that seemed useful to us in Franklin County. And each species, each plant species, each native plant species, was given between 1 and 3 of these descriptors. And then for the analysis, we looked at every species that contained a particular descriptor. So everything in this column has the aquatic descriptor. And we compared the change index in that group to all other species that did not carry that descriptor. And every place that you see an asterisk is a place where the species carrying that habitat descriptor differ significantly from the other species. And you can see that actually most of the habitats do show a significant difference. Now, here's where there are challenges of interpretation. For example, the biggest apparent increase here is in aquatic species. And my colleagues and I would argue that this is probably virtually entirely an artifact of sampling. Certainly, herbarium specimens in Franklin County have been collected from aquatic habitats. There are several hundred of them. But if you look at those specimens, they were rather focused on a relatively few bodies of water. Ashfield Lake in Ashfield, Leverett Pond in Leverett, Lake Wyola, formerly Lock Pond in Shutesbury, and a few others. Whereas we tried to be very systematic in our sampling, and we tried to cover ponds in every single town. And I should say more specifically, Matt Hickler covered ponds in every specific town because he was the aquatics man in this group. So I think really this is an artifact of the fact that we, and as I say, it's Matt, got around to aquatic habitats in every town. Now, take something on the other end. According to this, ledge species have a significantly less positive change index than the median. So does that mean ledge species are decreasing relative to other species? It could, but there's also another possible interpretation that is tied more to sampling. Franklin County has some interesting ledges. It has limestone, albeit dirty limestone. It has basalt. It has sedimentary rocks like the Mount Toby conglomerate that seems to have some calcareous elements or base-rich elements to it. And the outcrops of these rock types were well known to botanists in earlier days. They were sites of unusual ferns. The talus slopes in nearby areas often were sites of interesting orchids and other rich woods species. And we suspect that quite possibly these ledge habitats got visited more heavily than did other habitat types back in an earlier day. And perhaps they're not decreasing at all. Notice, by the way, that the mean here is actually above 0. So that means we're finding ledge species in just as many towns, actually a few more towns than they found them back then, but the increase is not quite as much as the median. So are we to interpret this as a decline in ledge species or a sampling difference? Okay, we can't know for sure, but we lean towards the sampling difference. Of all the significant differences here, the one that strikes us as the most likely to be real is this one, ruderal habitats, weedy habitats. Native plants with ruderal affinities show a large increase. And in this case, it is likely, in fact, that this habitat is increased. We have not done a detailed analysis on this for Franklin County, but I did do one in Worcester County using aerial photos between 1971 and 1999. And this was the habitat category that had actually increased the most in that period. It was a 2 to a 3-fold increase. And so an increase, and probably somewhat similar, maybe a bit less in Franklin County, simply because the population density is lower. But it's likely that there has been a real increase in ruderal habitats, and therefore it is no surprise that there might have been a real increase in species of ruderal habitats. Now, you might argue that, well, you're a little uncomfortable with this. You know, this is just kind of storytelling and finding patterns that fit our preconceived notions of what should be going on. And to a degree, I agree with you. I think that's the nature of the beast. But I would point out, or I would suggest, that simply a completely objective interpretation of the figures here representing real changes, I think that kind of approach would actually lead us to more errors than to try to bring our understanding of the plants, the land use, and so forth, and to make a more nuanced interpretation incorporating that information, which is what we've tried to do here. Okay. Another analysis that we did involved mycoheterotrophs, and it's possible this term is not familiar to you. You'll notice the root for fungi here, myco. These are plants that depend on fungi for at least some of their carbon. Now, you're well aware that most plant species have fungal associates that supply them with a portion of their mineral nutrients, particularly phosphorus. Mycorrhizae are the associations that undertake this. But mycoheterotrophs get a portion of their carbon. Now, there are various kinds of mycoheterotrophs. We have relatively few species that are full mycoheterotrophs for their entire life cycle. I've illustrated two here, Corollariza maculata, which is a non-photosynthetic orchid, and Hypophytis monotropa. I could have put in Indian pipe Monotropa uniflora. Those would be other complete mycoheterotrophs. There's only about 5 of those in the flora. But there are other species that are complete mycoheterotrophs during a portion of their existence. For example, all orchids are probably mycoheterotrophic during the time between the seed germination and when they start to photosynthesize. So you might call them sequential mycoheterotrophs or something like that. So orchids fall in here. Members of the Ophioglossacea fall in here because their gametophyte is non-photosynthetic and dependent on fungi for carbon. And also a number of the Lycopodium group also have a non-photosynthetic gametophyte dependent on fungi for carbon. In total, I believe there are 70 species in our flora that are some type of mycoheterotroph. And so a question of interest here, are mycoheterotrophs having more trouble than other species? And this is a plot. Here are our mycoheterotrophs here. Here are all of our other species. And you can see that there is a substantial difference here. This is highly significant. Mycoheterotrophs are not doing as well as other species. Now, this is just correlational results. I can't be sure that they're not doing well because they are mycoheterotrophs. Okay, it turns out this group is concentrated in a few families, the Orchidaceae, the Ophioglossacea, the subfamily Pyrroloideae and the Ericaceae, and a few others. And perhaps there are other things going on that simply happen to be correlated with mycoheterotrophy. Can't be sure on this, but as far as I haven't seen this pattern actually documented for any flora. So this maybe will encourage people to start looking into that. Okay. Families, plant families. Oftentimes there's a plot of which families seem to be showing the greatest losses. And I might point out that the Orchidaceae always shows up on this list, and notice that that one is highly significantly, it has a lower change index than the median of all species. And a number of other species that we see here regularly show up on these lists. The Ophioglossacea, the Galearis are the currants, ribes, and so forth. And of course, they were subject to an eradication effort because they're the alternate host for white pine blister rust. The Ranunculaceae often show up. The Violaceae often show up, perhaps less so these others. Now, keep in mind that we did this analysis on every family that had at least 5 species in it. And I think there are 60 some odd of those in the Franklin County flora. By chance, you'd expect 3 of them to be significant, okay? So it's quite possible that some of the significances that you see over in this column on this list are spurious, okay? But the one that really stands out here is the Orchidaceae, and we will come back to that in the third part of the talk. Okay, now a question that interested us here is, given climate change, given that the New England climate, the Franklin County climate has warmed over the past few decades, is there any evidence of this in the flora? Specifically, are northern species that reach their southern range limits in or near Franklin County, are northern species declining relative to other species? To get at this question, we needed to have some way to decide whether a plant should be considered northern or not. And we made use of the data in this Atlas of the Flora of New England, which is available online. And it has dot maps of the occurrence of each species in New England. For the most part, these dots correspond to counties, but some of the larger counties up here in northern New England have been divided into 2 or 3 parts. I'll refer to each of these units as a county just for convenience. And what we did here is we looked at the northern New England states, and there are 48 of these counties in northern New England. We looked at the southern states, that is the states south of Massachusetts, and there are just 2, and there are a total of 14 counties between them. We calculated the proportion of the counties occupied in the northern tier of states, the proportion of counties occupied in the south, and we simply subtracted those 2 numbers. So for this species, Gentiana linearis, 69% of the northern counties are occupied or have been at some point. Only 1 of the 14, 7% of the southern ones have been occupied. And you subtract those 2 and you come up with this number, call it 0.62. Anything with a number close to 1 maximum you could have. A number close to 1 indicates a northern species. A number close to -1 indicates a more southern species. And things that are near 0, neither one nor the other. So the northern species obviously are those with numbers closest to 1. We chose 0.4 as a dividing line. There's nothing magic about that number. It was used in the study of the Worcester County flora, but also it tends to correspond with our sense of things in the field. That is, if it's a species that we tend to find in cool microclimates in Franklin County, we like to see it on this list. If it's a species that is pretty widely distributed in the county, we don't like to see it on the list. And 0.4 seemed to be a pretty good dividing line separating those 2. Okay, well, the question then is, are northern species behaving any differently from other species in the county in terms of abundance change? This slide just illustrates a few examples with their latitudinal index. So these would be northern species. They all have numbers above 0.4. These are southern species with numbers below minus 0.4. Okay, so here is a comparison of what's going on in northern species compared to what's going on among all other species. And in this case, you see that northern species, a larger percentage, over 60%, are decreasing, whereas a much smaller percentage of other species. This difference is highly significant, and if we analyze it in a different way with Mann-Whitney, it's also highly significant. Northern species are doing less well than other species. So, are we done? Maybe, maybe not. In particular, perhaps why we're seeing a difference here is not because this group is northern and this group is not northern. Perhaps it's just that these northern species are range margin species. So they're not going to be as common, perhaps, as other species and may not do as well. And it may owe to the fact that they're a range margin species rather than specifically that they are a northern species. So can we get at that? Well, how about if we compare northern species instead of to all other species, we compare them to southern species, because southern species obviously are range margin species also. And that is in the next slide. Notice the pattern doesn't change very much. Large percentage of northern species is decreasing compared to southern species. The difference is, although the significance is not quite as high, they're both significant, the tests that we do. So these results also suggest are consistent with northern species having a harder time. But have a look at these sample sizes here. Notice our northern sample is quite a bit smaller than our southern sample. So maybe we should set a different cutoff for southern species so that we kind of get the same size of tail of the distribution there. Perhaps we're getting things that are approaching more the middle of their range in this larger sample. So I did one more analysis. And in this case, I compared the northernmost 5% with the southernmost 5%. Notice the pattern is similar. Higher percentage of northern is decreasing than southern, but notice the differences are not significant. Okay. In ecology, this is known as doing one test too many. What are we to make of this? We certainly have some evidence that northern species are not doing as well, but then we have this slide that the differences aren't significant in the way that we might have hoped. Well, we decided, and I have to thank Matt Hickler for this suggestion, we decided to come at this an additional way. That is, how about if we just look at the historical sample for a town and we calculate the proportion of northern species in that sample, and then we look at the recent sample for a town and calculate the percentage of northern species in that sample and see if they differ. It turns out that the mean historical sample for a town contained 3.8% northern species. The mean recent sample contained just 3.1%. And if you compare these figures, the difference is highly significant. And if you look here, every bar to the left of this line represents a town with fewer northern species in the recent sample. Any town over here indicates a town where there are more northern species in the recent sample. But as I say, the preponderance of the weight is over on this side. And so these data do in fact suggest that northern species are declining when viewed over all of the towns in the county. Now, one more follow-up slide on this. If northern species are declining in Franklin County, there are 26 towns in the county. They differ quite a bit in elevation. Where would you expect to see the greatest declines? In the higher elevation towns or the lower elevation towns? Seemed to us that it should be in the lower elevation towns because those are the places where the northern species are going to be closest to their range limits and might be having the hardest time. So we did yet another analysis where we looked at loss of northern species as a function of elevation. And notice there is a significant relationship here where the greatest loss of northern species is occurring in the lowest elevation towns and the least loss is occurring in the highest elevation towns, which is what you would predict if climate change is responsible for this. Now, one question may be occurring to some of you who are familiar with Franklin County. The lower elevation towns are in the Connecticut Valley, and those are the towns that have the highest human population densities and the greatest human effects of human land use on the, effects of human activity on land use patterns. Therefore, perhaps that's where we would expect to see the greatest losses of northern species, and it has nothing whatsoever to do with climate change. Well, we tried to address that by the way we coded this variable. This is not simply loss of northern species, but it's how much more loss of northern species there is in a particular town compared to loss of other non-northern species. So this is the excess loss of non-northern species. Human development, we would expect, would influence all species to some degree. So here we're just looking at the excess loss of northern species. And so I think that largely addresses that concern. I might also point out that a lot of the development in those towns actually occurred pretty early. Of course, Deerfield was one of the earliest towns settled, and a lot of the development occurred probably before many of the historical herbarium specimens had been collected. So in any event, I'm going to end this piece on Franklin County here. What do we conclude about the data on climate change? I would say that the results here are suggestive that northern species are in fact having greater difficulty than southern species. Okay, now part two of this talk, I would like to, and by the way, if any of you have any questions, I know you're not being allowed to interrupt at this point, which is the way I like to do it if we're in person, but feel free to include them in the chat so you don't forget them, and then we'll take up as many as we can at the end of the show. Okay, next question is actually going to be an elaboration of this last question that we were asking about climate change and northern species. It seems to me that if there is an effect here, it should be obviously much more broad than just Franklin County. And so I worked with a student at Holy Cross on this issue among state-listed species. We chose state-listed species because there is good recent regional coverage of those species through the natural heritage programs. And I would, whoops, here's the introductory slide for this, latitude changes among listed species. And I would first of all like to thank these individuals without whom we wouldn't have been able to do this. Bob Wernerl here in Massachusetts, Karen Zyko and Rick Enser in Connecticut and Rhode Island. Caitlin Spind is the Holy Cross graduate who was and continues to work on this project with me. And so what we're doing here is we're going to be looking at a mix of herbarium data and also data from the natural heritage programs. And it's perhaps easiest to indicate what we did if I show you a slide for one particular species. This is Linnaea borealis, a relatively northern species. Every place that you see green, or light green at least, is a town where that species was known from historical records, which for this study were pre-2000. Current towns with current records are in dark green, as illustrated here. Now, if these northern species are retreating northward, the average latitude of these current towns should be more northern than the average latitude of the historical towns. That would indicate a northward retreat. And you can see that in this particular case, it is. The recent latitude is actually about 2 degrees further north than the historical latitude, which is actually a pretty big change, okay? And you get some sense of that in looking at this figure here. So we did this kind of a calculation for a sample of about 25 northern species that are state listed. And then we also did the analysis for 2 control groups, which I'll illustrate in a moment. So here are the data on change in latitude of northern species. These are the species that we looked at. Everything that has a more northern recent latitude is in blue on the right here. The species that had a less northern recent latitude are listed here. In fact, if you analyze these, there is a significant tendency for recent latitudes to be more northern. And I might point out here, you might say, well, what about elevation? We actually included that in that when we calculated latitude, we normalized it for elevation. And so elevation has been taken care of in this analysis. So this group of northern species has moved about 0.7 degrees north on average compared to the historical period. If we look at the 2 control samples, this is a random sample. There is no significant difference between the historical and the recent. This is a southern sample, a very small change here, no significant difference. And so these data suggest that northern species may be in a bit of trouble, okay? Or at least they are suffering relative to other species. And they're consistent with the majority of the results that I presented to you from Franklin County. Okay, third little sketch here. Everyone's interested in orchids, okay? It's always exciting to find them, even if they're one of the most common ones, the pink lady slipper. We also know that many of them are quite rare. And there are many state-listed species, orchids. And as I illustrated in the Franklin County part of the talk, this is a family that seems to be showing a lot of declines, or at least many of the species seem to be showing declines. This is work that I have been doing with Karen Searcy. Karen has been focusing on more on the records from Franklin County. I've been focusing on the records from the 3 southern New England states, Massachusetts, Connecticut, and Rhode Island, to try to get a broader picture of what is going on. Our goals here, as far as I know, no one has systematically looked at the available data to see which species seem to be undergoing the most declines and which are not. So part of our efforts here are purely descriptive to find out what we can about changes. And as far as the purposes of this, perhaps twofold. Number one, it would be interesting if we can kind of establish the change of each species to see if there are any correlates with life history or environment that we can pick out. That is it predictable which orchids are undergoing declines? The other thing is that for many of us who do botanical fieldwork, I've talked to different individuals at different times, it seems that some orchids might actually deserve a higher level of state listing than they currently have. Obviously, you do need some documentation to be able to move things around or put them in certain groups. And so information that we generated might be useful in evaluating the status of these orchids in the natural heritage programs. Okay, well, where can we get orchid information? Historic information, it turns out that orchids, as I will illustrate with some numbers in a moment, orchids are actually very well collected, and they're very well represented in among herbarium specimens. There are over 9,000 species of specimens of orchids in the Consortium of Northeastern Herbaria database. So they've been sought out by collectors. And as far as recent records go, it turns out there are quite a few orchids that are state listed. And of course, the state natural heritage programs keep detailed information on them. So we have good recent information from that source. And a second source of recent information, which isn't as extensive as we would like it, but we do have town level data now for 2 Massachusetts counties, Worcester and Franklin County. And although that's not a random sample of New England towns, it's a pretty sizable sample. It's about 25% of all the towns in Massachusetts are in those 2 counties. And I'll present some data on how representative they are in a subsequent slide. Okay, well, what can we tell by looking at the herbarium data? First of all, if orchids are undergoing declines, what are the possible causes? Numerous suggestions have been made, but unfortunately, in almost no case has there been a study to evaluate these. So these are more in the nature of hypotheses. Some people have suggested that it's really mostly a matter of succession. Okay, our forests are aggrading, they're becoming more mature, and orchids like a bit more light and so forth, and they are losing out. Some have suggested that our increased deer population may be responsible, and many of us who spend time in the field have run into orchids that have been munched probably by deer. Atmospheric deposition has changed. We are adding a lot more nitrogen, for example. Acidity has changed. We have introduced lots and lots of earthworms to New England that were not here before. What do? They break down organic matter. That is also the food for fungi, and orchids have an obligate dependence on fungi in their early stages of development. Some have suggested that declines in pollinators, which there may be relatively few pollinator species for some of these orchid species, that could be important. Some have suggested that collecting itself could have caused changes in abundance. And then some people suggest that the decline is actually more apparent than it is real. If you're looking at herbarium data, they were accumulated over a period of many, many decades. And in any current floristic study that takes 3 or 5 or 10 years, you can't possibly hope to kind of see as many things as were seen over the previous century or 2. So lots of possible explanations here. Let's have a look, first of all, at the herbarium data. And just to illustrate some potential difficulties with the data, I went into the consortium database and found out where the most orchid species had been collected, most specimens, and where the most species have been collected. So have a look at this slide. In terms of number of specimens, New Haven, Connecticut is number 1. Amherst, Massachusetts is number 2. Now, you all know that there are some fine academic institutions associated with those towns. And if you look down the list here, notice Mansfield, Connecticut, home of University of Connecticut, the home of Brown University, Williams College, Deerfield Academy. A lot of academic towns represented here. Well, perhaps you're thinking what I'm thinking, that the founding fathers of these fine institutions decided to locate their institutions in towns with high orchid abundance and diversity because of the salutary effects that orchids have on higher education. Of course, there are other possible explanations. Maybe orchids tend to be seen where there are people to see them. Okay. And by the way, it's kind of interesting here. Southern New England has about 50 orchid species. So 2/3 of those have been found in each of these top towns here. Okay. Which is kind of interesting. In any event, clearly, the sampling is not randomly distributed spatially. How about temporally? Here are data for one species of orchid. It might not look too different for other species. These data are aggregated in 20-year intervals. And notice the peak in collection, 1910. We have a low around here and another low here. So if we were to take this at face value, we would say this species reached a peak of its abundance in 1910, a low at the time of World War II, and it's currently gone extinct. Well, I know it's not extinct because I took this picture over here in Monroe just last year. Clearly, intensity of collection has changed, and thank goodness people are not collecting as many orchids as they did in the past. Many of them are listed, and I think people just have a sense of a little bit more care with the collections. By the way, pay attention to this number. This is the sample size. I want to show you a couple of other slides just for reference. Another spring flowering wildflower. Peak abundance 1910, lows World War II, and in the present decade. Maianthemum canadense, peak collecting 1910, lows World War II, and in the current decade. And by the way, Maianthemum canadense probably has to be about the most common herbaceous plant in southern New England, certainly in the woods. Did you notice the earlier figure was about 180 for Galearis? That means that about a third as many Galearis have been collected as Maianthemum. Okay, and the actual difference in abundance, wouldn't you say, is probably at a minimum several orders of magnitude and perhaps more than that. Okay, clearly herbarium collections are not random with respect to almost any variable you can name. However, in aggregate, they're probably fairly thorough historically, just because they were sought out. Well, how about the recent data? Okay, as I say, the recent data for listed orchids can come from natural heritage programs. But for those that aren't state listed, we only have, as far as I know, Franklin and Worcester County to fall back on that give us town level records over a large area. So it's interesting to know whether what's going on in those counties is the same as what's going on in the rest of the state. And although our analysis will eventually incorporate the 3 New England states, here I'm focusing mostly on Massachusetts. Here is the percent loss of a number of orchid species in Worcester and Franklin County. Here is the percent loss in other counties in Massachusetts based on natural heritage data. Notice that there is a strong positive correlation between what's going on in Franklin and Worcester County and elsewhere. And if we remove this troublesome point that is based on just a small sample size for that species, the R value comes up to about 0.89, really a very strong correlation here. So this suggests that as long as we bear in mind that these 2 counties are not representative, they can at least give us the first approximation of what's going on elsewhere. So let's look at some data for a few species here. These are the ones that show the greatest apparent declines in Massachusetts. First of all, there are 2 species that are considered historical in the state, 100% loss. Where you see figures in parentheses, these are data that are based on Franklin and Worcester County and extrapolated from those. We have 2 other species that have very high levels of loss, a number of other species, excess of 90% loss. And by the way, 90% loss means that we only see it today in 10% as many towns as it was seen in historically. Now, not surprisingly, most of the species on this list have a designation of E for endangered, T for threatened, okay, or H for historical. These are the ones in the most trouble. But notice these 2, okay? These are 2 species that are just watch listed, but were never seen in the floras of Franklin and Worcester County. Okay, here's a species that isn't even a watch list species that is in Worcester and Franklin County gone from 90% of its towns. Okay, so there might be some candidates for some reassignment here. Looking at the species that have the least apparent declines, No surprise here. Okay, this is probably the most familiar orchid to most people, and I think most of us consider Goodyear pubescens and Spiranthes cernua to be two other most common orchids. Notice, by the way, that here are two endangered species that show relatively low loss. Of course, these aren't known from very many sites, which is why they're on the endangered list. And this one, Isotria medialoides, is one of the few federally endangered species that occur in Massachusetts. Okay, maps are always fun. Let's take a quick look here at a few species. Platanthera flava, a threatened species. Light green are towns with only historical records, dark green with recent records. Liparis liliofolia, notice that it is no longer present in eastern Massachusetts. It seems to be largely restricted to towns in or near the Connecticut River Valley. This special concern species, there are not so many light green towns here. And in fact, I'm going to show you another slide of this species where I've separated out the recent records into towns that had a historical record. So both means that in this town, there's both a historical and a recent record, but the pink towns are only recent records, which for this study is defined as 1996 or later. So this is a species which is considered threatened that actually is being seen in a lot more towns today than it was seen historically. So what is going on here? Are we seeing an actual increase? Was this something that those sharp-eyed botanists of yore didn't see? Hard to believe, because there were some very good botanists back then. Not quite sure what's going on here. This is a watch list species. Notice the number of open circles in Franklin and Worcester counties. Okay, it seems this species might merit a higher listing. And again, if there are some of you that know of patterns in other parts of the state or southern New England that are very different from what I'm describing, I would be very interested to hear these. Platanthera hookeri, a watch list species. I've now been involved in 2 floras that have taken 25 years of my life. I have never seen this plant, okay, but it's only a watch list plant. Here's a couple of other watch list plants that I would argue are potential candidates for listing. Take this Platanthera here. Although you see 4 towns where we had recent records in Franklin County, the total number of plants in these towns was, I believe, only 8 or 9. So that's 8 or 9 known plants in a county, okay? And Platanthera orbiculata also doesn't look like it's doing so well. Now, this is a species, Spiranthes lacera, typical of kind of dry grasslands, that is not even a watch list species. And notice how many open circles. This is the species that there are only 10% as many recent records as historical records, okay? This might be something that we want to keep our eye on, at least informally. And Calopogon tuberosus, perhaps don't think of as particularly uncommon, but notice between these 2 towns how many open circles we have. It seems it's in a lot fewer places than it used to be. And then this is a species that I would argue doesn't seem to be showing much in the way of a pattern of change, Platanthera clavellata. It seems if you look hard enough in the right habitat, you tend to find this species in most towns. Okay, well, I mentioned this, we're at the very early stages of this investigation, and one of our goals is to try to tie how much change is taking place in orchid species with aspects of their biology. And we really have not started this analysis, but just this week I put together some figures on pollination. I divided the pollinators into 3 groups. There are some plants that are autogamous, they don't need a pollinator. Others that are pollinated by bees, and others that are pollinated by other pollinators, mostly moths, actually. And you can see that there is a difference in how much loss is being seen in these different species. The difference is, if you analyze it, statistically significant. Although, as I say, this is very early result, and we need to we need to be a little more sure of ourselves before we move ahead with, but at least suggest something interesting. Okay, I have talked long enough here. Let me summarize conclusions. I don't know that I've shown too much that is new in this talk. My hope really was to talk about ways in which we can use floristic data to assess change and some of the difficulties and pitfalls in doing that, okay? We have these various sources of information. Each of them has advantages, but each of them has potential problems in their interpretation, and we have to be very careful in sort of parsing out the real changes from those that are spurious. Increases in non-native relative to native species, that's pretty clear here, and I would argue that it has some important consequences for our native fauna. Certain plant families, especially the Orchidaceae, show high levels of decline, and I would argue that some of these orchid species are candidates for higher levels of monitoring or protection. Mycoheterotrophs are declining relative to other species. That will be something that will be interesting to pursue in other studies. And I would argue that the totality of the evidence that I presented suggests that northern species appear to be declining at the southern edge of the range in a manner that is consistent with effects of climate change. So thank you so much for your attention. I will stop sharing now, and when we get it set up here, I'd be happy to take any questions that you have.
All right, thank you very much, Robert. That was fascinating. Really, really great to see this overview of several of these projects you've been working on over the recent years. I would invite those in the audience to post questions into the chat function. And I will relay those to Robert in sort of a question and answer session here. While people are thinking and typing, I just also wanted to point out that behind that, the first section of Robert's talk is the recently published Franklin County Flora, a special publication of the New England Botanical Club, and that PDF of that publication is now available for free on the NEBC website. And you can also purchase hard copies of the Franklin County Flora on the website as well. So if you want to see more of the raw data behind that portion of Robert's talk, that is available through the Franklin County Flora. We had a couple questions come in during your talk. One was from Caro back at the beginning in the first segment about the Franklin County Flora. And she's sort of asking, there's a lot of analysis that you talked about of the northern species. And she's wondering if you looked in similar detail at the southern species and whether there's evidence of them expanding or increasing in abundance in Franklin County.
Let's see, I am unmuted. Okay, an excellent question. I did look at that question in Worcester County and looked at it also in Franklin County, a somewhat different way. It seems that southern species, at least in some of our analyses in Franklin County, are not doing as well as other species also. So they're a bit like northern species, but not as much. The simplistic sort of pattern that you might expect from climate change is the northern species would retreat northward and the southern species would expand from the south. But that is not obviously happening. And it seems to me that there could be some good biological reasons for that. For an extinction to take place, all you need to have is conditions that are unsuitable for that species. And those, if climate change is enough, could happen abruptly. Okay, species could just disappear. But colonization or expansion of the southern species depends on things like dispersal. And those are not instantaneous, particularly if you're an ant dispersed species and you have barriers to cross. And so it seems that a different set of processes will govern the expansion of the southern species compared to the retraction of the northern species. And I didn't show it here, but in Worcester County, I did an analysis, and for northern species, the towns they were disappearing from were in fact the towns at the southernmost edge of their range, which is where you'd expect if climate change is a key factor. The southern species where they were adding were not necessarily towns near their existing range. Rather, I was finding them kind of in infill towns that were within their historical range. And to me, that means they were probably there all along, but they were just overlooked. So bottom line, no, we did not find expansion of southern species. In fact, if anything, they seem to be having a little trouble too. They're also range margin species. And they definitely were not, they were definitely not expanding. You know, maybe you could find one or two species that you could make a case for, but for the most part, no. And I would expect a slower process.
Yes, no, that makes a lot of sense. There was Russ also had a question about the orchid portion of the talk, and was wondering if you looked at Epipactis and how that, the non-native orchid, and sort of how that compares to the native orchids, and what, to some extent, what might be going on there. It seems like that is fairly common. If there's any differences, maybe in its biology or ecology, that could explain its success as some of the native orchids are declining. And whether or not that was included in your analysis was part of it.
I actually have not looked at Epipactis because I didn't want to be distracted, but that you could imagine would be a very, very different pattern of collection times and changes than all other native species. And I would dearly like to know what Epipactis has that makes it so successful as an invader, if you want, that our native orchids don't. And I really don't have any idea. I have never seen anything suggested, although I haven't specifically read into that literature. And if anyone has any ideas on that, I would be very interested in hearing them. So I'm afraid I can give you no satisfaction.
Yeah, but it is a strange and fascinating exception to some of these patterns. Another question here, Katie asked about, in one of the charts you were showing of changes in northern species, Whately stood out as a sort of odd little increase in northern species. And if there's anything you can recall about the sampling there or unusual locations there that might have contributed to that.
Yeah, good question. I also observed that it sticks out like a sore thumb. It meant I had to expand my axis on the right side of the graph there. And I don't know, and I haven't really had a chance to discuss that with my colleagues, some of whom, including Jesse, is much more familiar with Whately than I am. I don't, you know, you might think, well, could the historical collections for whatever have avoided the cooler parts of town? But that's not obvious, because I would consider a place like Whately Glen to be one of the, maybe a slightly cooler microclimate, higher elevation. It's kind of down in a ravine. And that was, as far as I could tell, was well collected. So I really don't know. What I should do there is go and look at the particular species that we found that were not found historically, and see if I can figure out if there's a pattern in terms of habitat or a particular part of town. And I have not done that yet, but it would be a very useful thing to do.
Yeah. There's a question here in the chat from Jan asking about whether there's any attempts, or you're thinking about any attempts to define uncertainty around the change indices. So any sort of like resampling techniques or bootstrapping to sort of bracket those estimates.
I have not done that, and if I were better at this stuff, I probably would have. We have done, and this is not quite answering your question, but we've done an awful lot of analysis of one type or another for this. For example, one of our concerns here was that species that have different levels of commonness are constrained in how they can change. If there are 26 towns in Franklin County and red maple was collected historically in 25 of them, well, you can't have a very positive change index for red maple. It wouldn't even be as high as the median. On the other hand, a species that's only been collected historically in 1 or 2 towns could have a much more positive change index. And so at one point, we tried to normalize the change index for every single species by how many towns that species had been recorded in historically. It turns out the result, it was a bit of an exercise. It turns out the results that we got were not very different from the ones that I showed you with a more simple approach today. So that's why I didn't talk about them at all. So no, I'd have to give that a little bit more thought and probably talk to somebody who's a bit more accomplished in statistics than I am. Thank you for that suggestion.
Right. And Karen asked about the percent of land in the county that was sampled and sort of how you approached the fieldwork part of this in terms of, was it random sampling, random destinations, or was it targeted to public or land trust lands? And whether that, sort of how the surveys were targeted might have affected the results.
Good question. Although as an ecologist, I'm sort of well aware of and knowing a little about statistics, I am well aware of the desirability of random sampling and so forth. For a project at this scale, we just decided that was not practical. What we did is we sampled by town. Each town was visited at least once in every half-month interval between, I think, April 15th or May 1st and October 15th. We tried to visit as wide a range of habitats in each town as we could. We actually printed lists of both what we had seen in all towns and common species that we had not seen in particular towns. And we used those to guide our search so that if there were a lot of wetland species on the list, we tried to visit wetland habitats on the needed list. We tried to visit wetland habitats and so forth. So basically it was sort of driven by, I don't know, our botanical intuition and also by a desire to cover the towns systematically. We also, that is, if repeated visits to a town were turning up very little that was new, we tended to put more emphasis on those towns where we were still generating a number of new species, the idea being that those towns probably had not been as thoroughly sampled. So that's the best we can do there, okay? So no random locations chosen from maps or anything like that. Interesting locations chosen from maps is the way we did it.
And that, I mean, that sort of raises the question too of what we can sort of imagine the early collectors were doing and how they were conducting their, field work and their surveys. And that probably would also be sort of somewhat haphazard, somewhat targeted to interesting habitats. So in that way, somewhat comparable, but
well, we actually have the insight we have into that is actually the numbers of herbarium specimens collected in different towns. And that was highly, highly skewed in Franklin County. Deerfield has had an abundance of collections. Bernardston has had almost none. So certainly the historical collecting was haphazard. And as far as I can tell, there was no systematic attempt to cover all the towns and so forth. I will point out that Professor Alfred Goodale at Amherst College had done quite a bit of work and actually had a draft of a manuscript on the flora of this region, I think dating to the 1940s, that we have a copy of. And so I guess he felt at that time that the county had been examined enough that it justified a flora where they actually talk about which particular towns things are found in and so forth. That was never actually published. But I would say, no, ours was systematic, but not, kind of highly controlled as you would have like a plot level ecological study. And the historical sampling, unfortunately, was less systematic than that.
Could you say a word or two about the historical record in this in this area for Franklin County? What were some of the earliest specimens and collectors that were active in this area? And when did collecting begin in Franklin County?
I would refer you to a paper by Karen Searcy and Matt Hickler on a very important early collection, which actually is at Old Deerfield Village. And they published a note on that in Rodora. Those collection, that herbarium dates from the late 18 teens. Mm-hmm. And there were a couple of doctors that collected at that time. So the earliest collections were actually more than 200 years before our latest collections. So there was a little pulse of collecting there around 1820. And there were a number of other pulses along the way. There was a big pulse connected with the founders of the New England Botanical Club in the early 1900s. There was good ale in the 1930s. There was a remarkable woman, Roberta Poland, mm-hmm. Who collected extensively in Deerfield, and we always felt that we could never do quite as well as Roberta could, and she didn't even drive a car. So it was a bit episodic, but it took place over 200 years.
Mm-hmm. Great. There's a question here from Lisa about changes in aquatic species that you had seen, and wondering if there might be any connection with changes to pH due to atmospheric deposition that could feed into some of these.
Yeah, that's a very good question. And I don't know if Matt Hickler is around and wanted to unmute and address that. As far as I know, we don't have any evidence pointing to that.
Yeah, and Lisa also had commented that one potential source of bias would be that historical collections might be biased against common species, such that past collectors are going out searching for the orchids and ignoring Maianthemum. And she says in Natick, there's no historical collections of white pine or red maple in her town.
And she's sure that they were there.
Huh? Yeah, or maybe they're new. Maybe they're new.
Yeah, there are all kinds of biases certainly with the historical collections, and that's one of the things that makes this very, very challenging to try to parse out the real changes from the spurious ones.
Mm-hmm. We had one more question here from Russ, asking about those trends in sort of collecting intensity that you were showing. And there was this sort of distinctive dip in the 1950s, or maybe that's the 1940s to 1950. And then in the most recent decade from around 2010, any thoughts on that recent dip? Like what has been going on in the botanical community in New England such that the 1990s we're seeing more collecting than 10 years ago or currently?
I would say a couple of things here. First of all, you noticed that there was a dip in the most recent 20 years for Trillium and for Maianthemum. Mm-hmm. And so it's well known that collecting specimens for herbaria is way down, and looking to the future, that means the records from now are not going to be as helpful as they were the records from the past. So that's a factor influencing all species. However, I would guess with regard to orchids, number one, if we have 50 species of orchids, I would say in Massachusetts, just off the top of my head, there are probably 10 to 15 that are state listed, and then a number of others that are watch list species. And even for those not on either of those lists, as a collector, I almost, I don't know, I have a great deal of difficulty putting orchid in my plant press, so I think there's just a bit more, I don't know, respect for the species, or if you want to call it. And so I think people are much less inclined to rush around collecting these things today.
Mm-hmm. Great. Well, thank you very much. If anyone has any other questions, you can certainly drop them into the chat, and then we will also keep the Zoom open for a little bit more informal conversation at the end here if you want to stick around and ask any questions directly to Robert, or just visit with friends and colleagues here that are on the Zoom tonight. I want to thank Robert again for coming out tonight and giving a fantastic talk, dealing with some of these really fascinating issues about floristic change in our region, and evidence of potential impacts of climate change on our flora and changes in some of the rare species like orchids. So congratulations, this is fascinating and important work, and we had a great turnout tonight because I think there's a lot of interest in these types of questions. So thank you very much.